I want you to imagine trying to take a crystal clear, extreme macro photograph of a rapidly vibrating guitar string.
Oh, wow. Yeah, that's a nightmare scenario for any photographer.
Right. The focal depth is basically non-existent. The target is just blurring across your lens.
Yeah.
But now, and here's where it gets ridiculous. Imagine trying to capture that exact same photograph while you are actively jumping up and down on a trampoline.
Right.
That nearly impossible physical scenario, that's precisely the barrier neuroscientists face when they attempt to observe live microscopic neural circuits firing in an awake, running animal.
The mechanical challenge there, I mean, it really cannot be overstated. We are attempting to observe functional dynamics in the CA1 region of the hippocampus.
Which is deep in the brain.
Exactly. And we're not just looking at cell bodies. This means looking at distal dendritic arbors. We're talking about structures that are less than a single micrometer in diameter.
A fraction of a hair.
Yeah, a tiny fraction. But the brain is a highly vascularized, living organ. It pulses rhythmically with cardiac output. It heaves with respiration. And this is the kicker. The moment a mouse transitions from wakeful immobility to active locomotion, the surrounding tissue just violently shifts.
Which brings us to the focus of today's deep dive. We are looking at a really incredible May 2026 paper published in Neuron by Gonzalez and colleagues.
It's a massive breakthrough.
It really is.
Sure.
They didn't just marginally improve imaging techniques. They completely cracked this movement artifact barrier. They achieved stabilized three-dimensional dual-color optical recordings in behaving mice.
And in doing so, they provided definitive proof that dendritic arbors are operating as independent, localized computational processors.
Okay, let's unpack this. We need to establish the baseline physical scale of this movement problem before we look at the solution. How volatile is the CA1 region during normal behavior? I mean, how much is it actually moving?
The sheer physics of the environment makes standard two-photon microscopy almost useless for continuous observation. Brain tissue naturally shifts anywhere from 5 to 10 micrometers every 500 milliseconds.
Wow.
Right, so when your target dendrite is a fraction of a micrometer thick, the surrounding tissue is literally swaying at a distance 10 times the size of your subject.
And doing it multiple times a second.
Exactly. You end up with dark frames, blurred transients, and utterly unusable data.
So the historical workaround for this, the old way, was to physically inject synthetic fluorescent beads into the extracellular space to serve as visual anchors. Sort of like placing motion capture dots on an actor in a movie.
Right, the ping pong balls on the spandex suit.
Yeah, exactly. So why did the Gonzalez team abandon that protocol?
Well, introducing synthetic beads fundamentally compromises the biological environment you're trying to study. First off, injecting foreign particulate matter inevitably triggers a microglial immune response.
The brain's cleanup crew.
Right. You end up with localized neuroinflammation, and that fundamentally alters the baseline excitability of the very circuits you are trying to measure. You're changing the system by observing it.
Which defeats the whole purpose.
Exactly. And second, these synthetic beads possess an incredibly high quantum yield. When the imaging laser strikes them, they absorb and emit just a massive amount of photonic energy.
Which leads to thermal damage in the surrounding tissue.
Precisely. You risk literally cooking the adjacent synapses. Furthermore, that extreme fluorescence creates a severe optical artifact, a blinding glare.
Like staring into high beams.
Yeah. If the bead is situated too close to the target dendrite, the photon scatter bleeds into your recording and just completely masks the physiological calcium or voltage transients. But if it's too far away, it fails to accurately represent the localized tissue drift.
So the mandate was clear. They had to find an anchor that was completely non-toxic, native to the environment, and optically distinct.
Which is a tall order.
Yeah. How did they achieve that without just flooding the entire region with fluorescent proteins and ruining the contrast?
They utilized an incredibly precise technique called dual single-cell electroporation.
Electroporation.
Yeah. Instead of using viral vectors, which, that would cause broad, non-specific expression across thousands of cells and totally ruin their visual contrast, they used microscopic pipettes to deliver targeted genetic payloads to specific individual cells.
Okay, walk me through that.
So they targeted their primary CA1 neuron and electroporated it with two functional indicators, a green fluorescent voltage sensor called ASAP 6.1 and a red fluorescent calcium sensor called JRGECO1A.
ASAP 6.1 and JRGECO1.
Yeah.
Okay, we'll dive into the functional dynamics of those two indicators shortly, but the critical innovation was what they did next, right?
Yes.
They took a secondary pipette and targeted a completely different neighboring cell.
Yes, exactly. They electroporated the nucleus of an adjacent cell with a distinct red fluorescent marker, specifically H2B-TD tomato.
And why just the nucleus?
By localizing the expression strictly to the neighbor's nucleus, they created a dense, highly stable, permanent tracking sticker. It's confined. It generates zero immune response, causes zero thermal damage, and provides a stark, high-contrast anchor point native to the tissue.
Having a biological tracking dot is elegant, but the tissue is still bouncing around at kilohertz speeds. I mean, the mouse is running. How does the scanning hardware actually keep the laser focused on that moving target?
The stabilization is handled by custom hardware. It's an acousto-optical 3D scanning microscope driven by an FPGA, a field-programmable gate array.
And this FPGA is fast.
Incredibly fast. It's designed to execute a relentless algorithmic correction loop in under 500 microseconds, less than half a millisecond.
Unbelievable.
The logic of this loop operates in 3 rapid stages: drift, detect, and direct.
Drift, detect, direct. I love a good mnemonic. Walk us through the mechanics of that sub-millisecond loop.
Okay, so during the drift phase, the system fires 3 orthogonal scanning lines intersecting along the X, Y, and Z axes directly over that glowing red neighbor nucleus. Immediately, it enters the detect phase. The FPGA executes a localized center of mass calculation on the returning photons to pinpoint the nucleus's exact physical shift in 3D space.
It finds the center of the glow.
Exactly. Finally, in the direct phase, the FPGA recalculates the coordinate origin and updates the scanning mirrors before the main imaging laser even begins its pass over the target dendrite.
So drift, detect, direct.
Drift, detect, direct. It reduces motion artifacts by over 2 orders of magnitude, effectively shrinking the blur radius to submicrometer levels.
Okay, let's consolidate this first major hurdle before we move forward. Bullet 1. The baseline mechanical movement of the brain makes standard high-resolution microscopy impossible. Bullet 2. Introducing synthetic fluorescent beads disrupts the exact circuits we are trying to study through inflammation and heat. Bullet 3. 3D RTMC hijacks a neighboring cell's nucleus and pairs it with a sub-millisecond correction loop, giving us a permanent, non-toxic anchor.
Perfect summary.
So, to you listening, consider this. Imagine you're tasked with holding a laser pointer perfectly steady on a sporadically moving target. If you could automatically correct your aim every half millisecond, what unseen microdetails of the world could you suddenly notice?
And with the tissue finally locked in place, the researchers could actually look closely at the functional dynamics propagating across the dendritic arbor.
Here's where it gets really interesting.
Yeah.
Once they had this kilohertz speed motion-corrected footage, they observed the mechanics of back-propagating action potentials, or BAP.
Right, the BAP.
We usually conceptualize the action potential as a forward-moving output, traveling down the axon.
Yeah.
But this data provides an unprecedented look at how that somatic depolarization wave washes backwards into the dendritic tree.
It's fascinating. The retrograde propagation of these signals is heavily dependent on the specific firing modality of the soma. The researchers categorize the somatic events into 2 types: single, isolated spikes, and rapid bursts.
And they behave very differently.
Completely differently. When a single action potential fires, the resulting BAP experiences severe spatial attenuation. The voltage degrades rapidly as it pushes outward into the arbor.
It runs out of steam.
Exactly. In the apical dendrites, the branches extending furthest into the superficial layers, the signal decays precipitously compared to the more proximal basal dendrites.
To contextualize this architecture, let's consider a sprawling multinational corporation. The soma represents the CEO, the central executive office, and the distal dendrites are the remote regional branches scattered globally.
I like this.
If the executive office sends out a standard low-priority memo, which is our single spike, the message loses its urgency as it travels across the physical distance. It passes through multiple management layers. By the time it reaches the most remote outpost, the signal is functionally dead. They ignore it.
The physical wire resistance of the neuronal membrane literally drains the voltage, just like bureaucratic layers drain the urgency of a memo. However, somatic bursts operate under a completely different biophysical mechanism. A burst doesn't just send multiple separate spikes. The rapid succession creates a sustained underlying depolarization envelope.
An envelope.
Yes. The baseline membrane potential elevates, and it remains elevated for the duration of the burst.
So the sustained envelope essentially paves the way. It allows successive spikes to push through the noise and overcome that physical resistance.
Exactly. Going back to your corporate analogy, a somatic burst is an urgent memo stamped in red.
Red alert.
The depolarization envelope drastically reduces the attenuation rate. The researchers quantified this decay curve mathematically. While a single spike's amplitude might fall below functional detection thresholds at merely 150 micrometers from the soma, the sustained envelope of a burst penetrates deeply into the most distal regions of the arbor.
The urgent memo reaches the entire company.
It reliably gets to every single branch.
Let's synthesize this second phase of the architecture. 1. Somatic action potentials rebound and backpropagate from the soma into the dendritic arbor. 2. Membrane resistance causes single isolated spikes to rapidly fade out over physical distance. 3. Rapid burst firing generates a sustained depolarization envelope, allowing the signal to overcome that distance and fully penetrate the distal branches.
Dot on.
Think about the way you process information. Have you ever noticed how a single isolated notification on your phone is super easy to ignore, but a rapid cluster of 3 or 4 pings instantly gets your attention? Your dendritic arbors seem to filter urgency using that exact same threshold logic.
They absolutely do. But the relationship between the soma and the distal branches isn't strictly top-down.
It's not just the CEO giving orders.
No. The RTMC system allowed the team to simultaneously monitor the central hub and the outer network, and it revealed instances where the dendrites were operating entirely off the books.
The localized independence of the arbor. The researchers documented what they called isolated dendritic events. These are moments where massive voltage transients were sweeping through the distal branches, but the soma remained completely hyperpolarized, completely silent.
The central executive was offline, but the regional offices were actively processing data.
Which is wild.
It definitively demonstrates that distal arbors are receiving, integrating, and calculating synaptic inputs locally. They are autonomous processing units. Furthermore, this autonomy is deeply modulated by the animal's behavioral state.
Right. What the mouse is actually doing matters.
Precisely. During periods of wakeful immobility, when the mouse is just sitting there, the soma and the dendrites exhibited high functional coupling. They tended to depolarize synchronously.
They were talking to each other.
Yes.
Yeah.
However, the moment the animal initiated locomotion, the moment it started running, that correlation plummeted. The dendrites uncoupled from the soma and began generating a high frequency of localized independent transients.
A major confounding variable here, though, is the sheer density of the network.
Right. Oh.
Because when an electrical signal decays along the arbor, or when a dendrite fires independently, how do we distinguish between network interference, synaptic noise from thousands of other active neurons, and the fundamental physical limits of the branch itself?
What's fascinating here is how they controlled for that network noise. It's a masterstroke of methodology. They used a highly targeted optogenetic protocol. They needed to bypass all spontaneous synaptic inputs and force the cell to fire in a vacuum.
To isolate the variable.
Exactly. To do this, they electroporated a specific genetically engineered light-sensitive protein, an opsin.
And this specific opsin was called R-Sharmine-M-Scarlet-KV2.1.
Quite a mouthful, yes. R-Sharmine-KV2.1. They targeted it exclusively into the soma. Functionally, this opsin acts as a microscopic light-gated ion channel.
So by directing a specific wavelength of laser light exclusively at the soma, they could manually trigger an action potential on command.
On demand, completely independent of whatever the surrounding neural network was doing.
They bypassed the entire brain's natural input.
Precisely. They isolated the variable, they manually drove the somatic spike with the laser, and then they measured the backpropagating wave along the arbor.
And what do they find?
The resulting decay curves perfectly match the attenuation profiles of spontaneous natural BAPs.
Wow.
This proves definitively that the attenuation is cell-intrinsic. It is not outside network interference. It is dictated entirely by a physical property of the dendrite's shape, the structural diameter, and the specific distribution of ion channels.
It's the wire itself causing the decay.
Exactly.
Let's capture this third paradigm shift for everyone. Bullet 1. Distal dendrites routinely generate isolated transients, acting as independent microprocessors while the soma is completely silent. Bullet 2. This localized autonomy dynamically scales with behavior coupling is high during immobility, low during running. Bullet 3. Using laser-triggered opsins, researchers prove that signal decay is a fundamental, intrinsic physical feature of the cell itself, not an artifact of background noise.
The methodology to prove that is just beautiful.
It really is. So think about a scenario in your own life where you had to execute a rapid, complex, physical reaction, like catching a glass before it hits the floor, without consciously waiting for higher-order cognitive approval. If your brain's peripheral remote branches didn't have this localized autonomous processing capability, could you physically interact with the world at functional speeds?
You'd be way too slow. Which brings us to the most complex dimension of this entire study, the final piece of the puzzle. We have to separate the electrical data from the biochemical data.
So what does this all mean? Up to this point, we've focused almost entirely on the voltage transients.
The electricity.
Right, the rapid electrical signals captured by the green indicator, the ACP 6.1. But this was a dual-color imaging study. They were simultaneously tracking the intracellular calcium dynamics using the red indicator.
The JRGECO1A?
Exactly. We need to distinguish the functional roles of these two mechanisms.
It's a critical distinction to make. Voltage is the rapid transient propagation of electrical information. It's just information passing through, and it occurs on the scale of milliseconds.
Fast and fleeting.
Yes. Calcium influx, however, is the heavy biochemical trigger. When voltage-gated calcium channels open, the resulting flood of calcium ions initiates a massive cascade of intracellular signaling pathways.
It physically changes the cell.
Yes. This calcium influx is the fundamental requirement for structural neuroplasticity. It physically dictates synaptic strengthening or pruning. It's the mechanism of learning and memory. So voltage is the message. Calcium is the structural adaptation to the message.
And when the team mapped both of these pathways simultaneously across the arbor, electricity and chemistry at the same time, they discovered a severe divergence based on the geometry of the branches.
The branch order.
Right. They classified the arbor into distinct branch orders. The primary trunk exiting the main cell body is branch order 0. A bifurcation, a fork in the road, creates branch order 1. Another split creates branch order 2, and so on, all the way out to the tiny twigs.
The higher the branch order, the more structural bifurcations the signal has navigated. And using advanced computational clustering algorithms on this dual-channel data, the researchers identified a striking physiological phenomenon in the distal regions. They called it the strong decoupled event.
Strong decouple.
In these high-order branches, they routinely recorded massive, high-amplitude electrical voltage spikes that were accompanied by absolutely zero calcium influx.
Massive electricity, no chemical change. To mathematically validate this decoupling, the team utilized a feed-forward neural network paired with principal component analysis, or PCA.
Yes, PCA is crucial here. They used PCA to reduce the extreme dimensionality of the imaging data, isolating the core statistical variances.
Cleaning up the massive amount of data.
Exactly. Then they trained the feed-forward neural network to decode the exact state of the soma strictly by looking at the distal dendritic activity.
Predicting what the CEO is doing based only on the remote office.
Precisely. And when the algorithm relied on the dendritic voltage data, it successfully and highly accurately predicted the somatic output.
It worked.
It worked flawlessly. But when they forced the algorithm to decode the soma using only the dendritic calcium data, the predictive accuracy collapsed entirely, especially at those higher branch orders.
Because they are decoupled. Let's visualize the mechanics of this decoupling. Imagine the distal branch as an industrial pipeline system. The voltage represents an electrical signal wire running alongside the pipe. The calcium, on the other hand, represents a massive, heavy industrial water valve that requires significant physical torque to crank open.
That's a great way to picture it. Deep in the proximal roots near the soma, the electrical signal carries enough shear kinetic energy to trigger the motor and open that heavy valve.
Yeah.
Voltage and calcium are tightly coupled there.
The signal wire opens the water valve.
Yes. But as you trace the network out into the high-order distal branches, passing through countless structural bifurcations, all those forks in the road, the electrical current physically dilutes.
The electrical signal, the voltage, still arrives at the distal edge.
Yeah.
The information is successfully transmitted.
Right.
But it has lost so much amplitude going through all those bifurcations that it simply lacks the biophysical torque required to pry open the heavy calcium channels.
The voltage spikes, but the biochemical valve remains shut tight.
If we connect this to the bigger picture,
the evolutionary efficiency of this design is just staggering. It allows the distal arbor to rapidly process, integrate, and forward massive amounts of high-frequency electrical data without indiscriminately triggering the calcium cascades that permanently alter synaptic weights.
it's protecting the structure from constant rewriting.
Exactly. It creates highly localized zones of neuroplasticity. Distal branches possess fundamentally unique, isolated biochemical filtering rules compared to the proximal structures.
Pulling together this final piece of the puzzle. 1. Those structural bifurcations of high-order branches physically dilute the electrical current. 2. This dilution creates strong decouple events at the distal edges, where we see massive voltage transients but absolutely zero calcium influx. 3. This decoupling establishes highly localized, geometry-dependent rules for synaptic plasticity and learning across different zones of a single cell.
Which is incredible.
It really is. So to wrap up this section, think about how branching splits the signal. This decoupling means learning mechanisms are completely different depending on exactly where on the dendritic tree the input lands.
This raises an important question, though, and it fundamentally challenges how we model neural networks in general.
How so?
Well, we are historically trained to view a single neuron as a binary node. You know, a single integrated switch in a larger circuit, it's on or it's off. But this stabilized 3D imaging data proves that a single neuron isn't just one switch. It is actually a central executive hub dynamically connected to thousands of autonomous, biophysically distinct mini-computers.
Thousands of them in one cell.
Exactly. If each microbranch filters electricity and chemistry differently based on its precise geometric location, and if each branch can execute localized calculations independently of the soma, how many simultaneous computations is a single human brain actually executing?
The numbers become astronomical.
They do. If we replace the concept of one cell, one node, with one cell, 10,000 nodes, does this completely redefine the theoretical computational ceiling of the human mind?
It exponentially shatters our previous models of biological processing power.
Yeah.
I mean, the sheer density of the architecture is breathtaking.
It's a whole new frontier.
It truly is. Thank you so much for joining us on this deep dive. I strongly encourage you listening to rethink your own cognitive capacity today. Be aware that millions of independent, localized corporate branches inside your head are actively filtering, calculating, and adapting without you ever having to consciously direct them. Keep learning, keep questioning, and we'll catch you on the next deep dive.